properties of polymerosomes through molecular dynamic simulations. Thank you, Monica.
Thank you for this opportunity to talk here about my work. In this time, I will talk about drug delivery systems. In particular, I will talk a little not, but about polymerosomes. But let we start. We talked in this biophysical meeting too much about biological things, especially focus on proteins, DNA, RNA, and membranes like a structure that that keep the cell and all the compartments. But the biological membranes have their own importance, and the molecules that biological membranes form the cell wall structures of all cell and cell compartments, contain a mix of different lipids, esterols, are site of actions of different proteins, and they are formed, the structural basis is essentially the lipid bilayer. The lipid bilayer is formed essentially for lipids. Lipids are particular molecules that have an hydrophilic head and hydrophobic tails. In water, they look to accommodate in some way that the head interact with water and the hydrophobic tails go out for water. This is quite a simplified picture, but it is important because lipids have a very important particularity that they act together. Although a lipid itself is a quite simple molecule, but they are a cooperative molecules that work together to make these walls and these structures. But as a physicist, if you have the cell as very complex system, you want to see what happens if I pick up the different components and try to understand each in separated way. In this sense, models of lipid membranes can help too much to understand this kind of properties. For instance, if you just take lipids, one or a controlled way to have lipids, to choose the lipids, you can, and you put in water phase, you will find different structures related to the shape of the lipids and this kind of thing. In particular, one that I want to call the attention today is the liposomes. Are spherical vesicles, concentrically organized phospholipid bilayers, separate but internal water phase, that are like a small sphere with a water inside, and the lipid heads interact with this internal water, and a hydrophobic region and a hydrophilic region. Well, what happened with this kind of mimetic system? This kind of mimetic system that also exists in some biological situation can be used for different applications, very, very different applications. One of them that I will put the attention today is to function as drug delivery systems. Well, why is it important to have a drug delivery system? Let's continue. Well, some of you mentioned the difficulty that is to find a new drug, and also the long way that you should go to find a new drug, validate it, do all the phases that we learn also in pandemic that you should go through when you have a new drug. And this long road takes too much money, too much time, too much effort, and most of drugs are not approved. Yeah? But Many of the drugs that are already in the market are good drugs, but they don't access to the right place or have problems to access to the right place. Then one strategy is develop nanocarriers that help the drug to overcome the problems that they have to reach the right place. For instance, we can, when we talk about a nanocarrier, we can talk about different kind of nanoparticles. It was mentioned in the talk before. The dendrimers could be liposomes, could be very different kind of solid lipid nanoparticles. There are many of them. That it depends of the application and the place where you should deliver the drug. Yes, for instance, if you need a particle that goes through the blood, go into the blood, you should put usually polyethylene glycol to go through this. But I will go again over a concept that Paul did yesterday. What is the ideal nanoparticle? The nanoparticle that is the spherical cow. Well, as I said, each nanoparticle will depend of the application that we need and the kind of special particularities that they have. But the ideal nanoparticle should encapsulate the drug, take the drug to the pharmacological target, release the drug, or slow or fast, it depends, but be completely removed and don't be toxic, yes? And which is the ideal nanoparticle will depend of each particular problem. But the question here is what we can learn from simulations, both at the molecular level, atomistic or coarse-grained model. Let's back to the liposome. See, we have a liposome, yes, we have a liposome, and we want to take some information from the liposome. We can do two things. We can take a very big liposome, radius, infinite radius, and take a slide here. Take a little slide, and suppose that you have a planar bilayer. That is a quite common approximation that we do. That is one way. Another way is to study a small liposome. A small liposome will give us some information about the effect of curvature, but usually we can access to quite small ones. Yes? And the reality, when you talk about drug delivery systems, will be around the hundreds of nanometers. Yes? So we can extract information from this approximation and from this approximation and can try to compare and see what can we get and go and back. Well, liposomes are quite good drug delivery for local anesthetics. This story started here in Brazil long time ago. I started to collaborate with Eneida de Paula, that she investigate everything about local anesthetics and entering the drug delivery systems. And there are a very particular interest. Local anesthetics are a drug that cause reversible local anesthesia. You know when you go to the dentist that you get the anesthesia. In 4 hours, you get 4 hours, you get free of this. Yes, but what happens if you, for instance, want to use the local anesthetics for the treatment of for chronic pain? You cannot put each 2 hours or 4 hours the local anesthetic. So the local anesthetic should be to treat the chronic pain should be active for a long period of time, should have low toxicity, and have good solubility in water. And this could be achieved using liposome, for instance. Why? Most of, I will say next, but most of local anesthetics have a pKa that is around 7 something, 7 or between 7 and 8, yes? And 7 and 8 means that at a physiological pH, they could be protonated or unprotonated. But you have both species. So liposomes are good species to carry both hydrophilic and hydrophobic drug. Well, first, we start to work quite a lot, and is our model drug, with prilocaine. Prilocaine is a local anesthetic that and we study in lipid bilayer both neutral and protonated anesthetics, and we carry out several molecular dynamic simulations, and we take the fingerprint that usually is the electron density profile or mass density profile. But we want to go to more complex systems. In this way, we go from atomistic to coarse-grained model, yes? And we use Martini force field. There are lipids, water, amino acids in Martini force field, but not this particular drug. How we did this? We mapped using some rules, and then we try to reproduce, atomistic simulations using this coarse-grained model, and you define tanning to get the same result or similar results that fit the atomistic simulations. This means that protonated, I will result this, protonated species go to the lipid-water interface and the water, and neutral anesthetics have a bimodal distribution around the inside the lipid bilayer, in the hydrophobic region. Well, we go to higher systems, to bigger systems to try to investigate this, and we put both kind of anesthetics in water in presence of a liposome, and we carry out simulations, and we find that all neutral anesthetics enter into the liposome, and protonated partition between both phases. Well, but try to mimic nature. We talk about liposomes that are these kind of particles formed by phospholipids that are amphiphilic molecules. What happens if we have other kind of amphiphilic molecules? Or we design amphiphilic molecules like D-block copolymers, for instance, that can have hydrophilic and hydrophobic regions, that we can have different units and can syntonize and have different length of each of the parts. Well, it was found in 96, 97 that they can build with this kind of polymerosomes, this kind of block copolymer, the block copolymer, they can make spherical structures like liposomes that are called polymerosomes. What we can do from simulations? Okay, we start from coarse-grained model. We map each unit, each monomer of the unit in a bid, and did a PO, polyethylene oxide in polyethylene butadiene. And we start from a completely messy simulation. We put randomly all the peptide, the block copolymers and the water, and we look at the self-assembly. And we found that we got a self-assembly of a lipid bilayer. of course, considering periodic boundary conditions. And then try to go to higher polymerosomes. But first, we also check how they are when we change the polymer length. And we found a quite stable lipid bilayers, polymer bilayers, for different lengths of the hydrophilic and hydrophobic tails. Well, next step was look for a drug. Look for a drug, and we have already a drug that we already mapped in Coursera. We said, okay, maybe will not be the right drug to put in a polymerosome, but could be a very good testing drug for two reasons. One, we have a hydrophilic, that is when the drug is protonated, and hydrophobic when the drug is neutral. So we go to study this kind of polymerosomes, and we found, as happened in liposomes, that the hydrophobic drug go inside the polymer bilayer. And here we have the fingerprints that are the mass density profile in this case. We see that differently that happened in liposomes. Here we have a more uniform distribution of the hydrophobic drug. What happened with the hydrophilic drug? When we put the hydrophilic drug, they partition between the interface and the water. Here I am showing a picture. You don't take conclusion from the picture, but you see here that when you quantify this and put in the mass density profile, you see that have many of the drugs that are trapped in the interface between the hydrophilic and hydrophobic beads, and part of the drugs are in the water. Well, we change the concentration and estimate if they have some saturation, but then we want to encapsulate both drugs in, because at physiological pH we have a combination, both drugs in the entire liposomes, and compare with the lipid bilayer, with the polymer bilayer. Well, for this we calculate the pressure profiles. Bilayers have a planar symmetry, so we just suppose that the pressure in X and epsilon is the same, and we calculate the tangential pressure as the half of sum and half of n, and normal pressure as the anisotropy X. What happened with the spherical symmetry? Something very similar, but in theta and phi, but the anisotropy axis is the normal axis, will be the radial one. Well, so we come back and did the calculations with this. Here we compare the mass density and the pressure profile. We see here the pressure, the normal pressure, essentially it's didn't change. It's 1 bar, but the tangential pressure have changes when you have different interfaces, and it differs quite a lot from the polymerosome. Then there are mechanical things, mechanical properties that you are not getting from lipid bilayers because of cancellation and this. Well, we did the same with I will not enter in details, but we did the same with both drugs at the same time with bilayers and polymerosomes, and we get quite interesting results. So, I am in the last minute. For bilayers, we observed that the structural behavior of the system is guided by the protonated species, because they they are in the interface between the hydrophobic bits, and they expand the lipid layers. And this is frustrated in liposomes. You cannot expand as you want because you have different conditions. In polymerosomes, are more guided by the neutral species because the size, the expansion will be related with the drug inside the polymerosome. Well, we implement this method in a home version of GROMACS to calculate the profiles, both for planar and spherical. And now we want to, at this moment, I am moving from drug delivery systems, and we try to understand interaction of antimicrobial peptides with membranes. So in these cases, we can have different mechanisms of actions that some of them could be useful to use this kind of tools because you have a quite good distribution of the antimicrobial peptides. But if you have, for instance, a pore formation, you have an asymmetry that you cannot use, for instance, the approximation that we are using. Well, thank you all of you. Have collaborators in different stage of this. Eneida de Paula and Florencia Martini for the interaction of anesthetics with the lipid membranes, and Marta, Julio, and Damian and Juan. Damian and Juan were who did the work, and now this is my group in Buenos Aires. Thank you all for attention.
Thank you, Monica, for your very nice work. Questions?
Hi. So over here, it was really very nice, and it's actually on this slide that I want to do the question. So you actually, when you go to the polymerosome, because of the radial symmetry, you break the symmetry, right? Mm-hmm. Because of it behaves differently. How much of that should be kind of minimized when you go to like larger liposomes, more realistic, big? Large. So did you ever try to change the size of the polymerosome to see if you correct this?
When we did this calculation, we were in the limit. We didn't try it, but polymerosomes, differently that happened with liposomes, the effect of curvature is quite high. For this, I said in the beginning, we calculate one extreme and the other, and reality is somewhere in the middle. Yeah. Could be a very good idea, but I guess that have another problems, because when the we have a self-assembly or this kind of system are a mix of kinetic effects and energetic effects, because the number of the molecules that get inside is quite big. I mean, it's quite have a very big dispersion. Yes. For instance, when I will talk about liposomes. When you do liposomes, you do the mix, and you get many different sizes. Then you sonicate it. They do some process to get the right size. Yes, but in the self-assembly, will not. I guess this will be but I think that here we have also in the NEAT, I think we also are putting too much because of the ensemble that we use to do the calculation, NPT. So I don't expect that this converges in this, but I want to get more information of this, that is the system of the real system of interest, and with the radius get bigger. Well, I don't know if I answer your question, or we can discuss later. If not, I go. If they don't have another question, I continue. I come back to this.
Monica, I have a question myself. Yes, because we know that the membrane, the biological membrane, the composition of lipids in membrane is asymmetric in the inner and the outer surface. How difficult is to model in this kind of situation, especially when you have drugs? Because you can have different
the stuff that we
phases.
consider to build up, they are not symmetric. They don't have the same number. But we consider in lipids could be a little bit easier, and a little bit easier to do some tricks. We consider the lipid area and the lipid area of the outside and inside, and usually you have the thickness of the bilayer. For instance, one trick that you use with the liposomes is to keep an open hole and let the liposomes, the lipids, to enter and fill it as they need to finish. The thing that happened, and in the same, it's answer to you, another answer to you, these kind of systems are very, very floppy. So they don't have the right number. They can accommodate much more easily than lipid bilayers, but also varying the thickness. And in this way, if you vary the thickness, it's more difficult to estimate how many do you need for specific area. Yes? It's in this direction?
Yes, I was wondering how easy it is to simulate asymmetric membranes.
Yeah,
It is.
We use the information that we have from this to calculate the area per lipid or per polymer, and estimate the area that we will cover in the other. When you have a very small one, we extrapolate because you have the same head, lipid head, and the conformation of the lipid tails are not the same in one side and the other.
Okay, we can talk later, and especially making a liaison with the pH or the protonation that we can change through the bilayer.
Yes. It is a big deal. We in the beginning we decided. Well, in the beginning, no, we stopped there. But we decided to treat both cases separately. And then we put both species, calculated with the Henderson-Hasselbalch equation, at the work conditions, for instance, physiological pH. But one idea that was in there was do protonation, the protonation in the flight, because not necessary for the drug delivery system, that is also important, could be, for instance, pH dependent gradient to have much more protonated in the internal cavity and more in the, and the other in the, to increase the number of drugs that you can carry. But for the mechanism of anesthesia, they also seems also important because they know where they act, but seems like a neutral one can diffuse and protonated ones are the actives. Then this protonation, the protonation will be important for the biological role.
Okay. Any other questions? Then you thank you, Monica, again. Thank you. Thank you. Very nice talk. And I'm going to, right now, to call Professor Vitor. We changed the chair. So
Thanks, Rosangela. Well, now we are going for the final part of our meeting and our grand finale with José Onusiek. I think you all know him here, and he's actually responsible